I'm Patrick Penner, and I'm going to be talking to you about computing and drug discovery. Thank you. I haven't said anything that amazing yet. So I'm usually in pharma, and we do quite a lot of computing. I'm in computational chemistry mostly, and I will try to kind of put myself in the context of drug discovery and how we compute in that context in a moment. So, because we are recording this, brief disclaimer, my opinions are my own. None of my past or future employers have necessarily endorsed what I say. This is just me saying this. To give you a little bit of context who I am, what I do, so I have a master's degree equivalent, it's a German degree, in pharmaceutical sciences, and this is where I started, in the natural sciences, and only then did I sort of slip into computational stuff by learning to code on the side and then applying that to my work, which ended up in what is technically a doctorate in computer science at the University of Hamburg. It was mostly still doing the same things, so I did my master's with my professor Thierry Lange at the University of Vienna. Thierry Lange had a software company, had co-founded a software company at the time, and some of what I did in my master's flowed into that company. And I did also do an internship at Böhringer at the time at the University of Hamburg. I worked with Professor Matthias Raray, who was my doctoral advisor, and there we were in collaboration with Servier Pharmaceuticals and Roche, which ended up in me then eventually going to Novartis Pharmaceuticals. And I'm saying this not necessarily to flash you with all of my professional experience, but to show you that drug design tends to be a corporate sport. We are generally fairly close to companies, and I have been very close to companies my whole academic career and my professional career. So on one hand that, and on the other hand I do want to show you what a computational chemist looks like. Usually we do come from the natural sciences and drift into computer stuff. It's fairly rare that you have computer scientists going the other way, for the obvious reason that you could just get a job just with a master's in computer science and then don't necessarily do a doctorate or anything like that. And this is very complex data that we're working with, and so usually you don't really have people with the patience to get into that, and so you can just get a nice job in programming something, right? It's usually people from natural sciences drifting over, as I did from pharmaceutical sciences into some more computational aspects. I want to talk about computing drug discovery in these 5 sort of areas, starting with the data, giving you an idea of what we're actually working with and how we actually put chemistry in computers, which was a fundamental question for me, definitely. Some mathematical modeling we do in the context, some structure-based modeling we do in the context. This is the real 3D stuff of 3D proteins and 3D molecules, and we'll see a little bit about that. Some computing techniques we use. We are heavy HPC users, and I want to talk about some of our compute loads, as well as some organizational challenges, and I think it's the last 2 that may be the most relevant to you. If you ever have customers, or if you ever deal with people from this sort of an area, those are probably the things that, in an abstract way, may influence you. So, one more context slide. Drug discovery is a very, very large process, and I very intentionally named this talk Computing and Drug Discovery and not in development or anything like that. In discovery, I have spent most of my professional life halfway through here, the target to hit range, to maybe halfway to preclinical, seeing preclinical results once in a while. The rest, I spent 4 months generally in clinical stuff, but not a lot. But it's really this target to hit and no, we're not going to talk about anything preclinical today, but target to hit to lead optimization that I'm going to be talking about. What does that mean? So in the process of how we make a drug, we start with a target hypothesis, and this is usually very, very basic research by biologists that do unspeakable things to mice. Some of that they justify with modeling disease, and sometimes someone in industry actually reads those papers and sees, hey, maybe this is real, maybe this is something that we can make a drug with, suggests it to their superiors, and then they reproduce those unspeakable things they did to mice in the company itself, see, okay, is this real? And if it is, you maybe go on to the next step, which is the target to hit. You have maybe found a protein that might modify disease. Let's try and hit it. Let's hit it and see if we can really modify disease by hitting it. Hitting it means in my context, especially in computational chemistry, means finding a molecule that influences its behavior. There is a whole other aspect of doing this with more biological molecules, with larger molecules, that I won't be talking about today. I will be talking about the chemical applications. Some of these techniques go across these disciplines. I will be focusing on chemical molecules modifying biological behavior. And so this target to hit block usually involves either you taking the molecules that are already known to influence a particular protein's behavior, a particular signaling pathway's behavior, and just working with that, going with that. If you believe in those enough, you can go with that. Otherwise, we often also run huge screenings of millions of molecules to see, okay, can we find any kinds of molecules that could really do something with this? But that's expensive, so we don't like to do that. So you find some set of molecules that you want to start to work with, and and that goes to hit to lead. These hits are usually very dirty, explicitly poisonous, and often enough not even water-soluble molecules. So even if you tried, you probably couldn't get that into a person just because it doesn't solve in water. And unless you're really putting it into them with a syringe, it's just not going to go into the person. And this is the hit to lead process where we try to solve these really, really big challenges of something that's not crazy obviously poisonous, and something that will actually go into a person and roughly does what we want it to do, which gives us a lead. A lead molecule is already a somewhat cleaner molecule, and in this lead optimization phase, we then fine-tune it. We fine-tune it with regard to what we actually wanted to do in the clinic. So, for example, you want to only dose a patient once daily, and you need a certain molecule to achieve that. You need a certain amount of solubility, you need a certain amount of efficiency of action for that molecule, and this is the kind of thing that you solve in lead optimization. So we're already looking towards the preclinical, and then preclinical starts, and this then goes on into human trials eventually, if the molecule in the preclinical or other phases does not turn out to be either super toxic or ineffective, which can still happen after that point. So we're talking target to hit to lead, lead optimization, around there. If you have any questions, please interrupt me. I've been told I have time, which is a very, very dangerous thing to tell an academic. But let me start with the data and the really fundamental question of how do we even get molecules into a computer? And this was the question I asked myself in the 6th semester of my pharmaceutical sciences degree when I had learned to do basic Python stuff. And I had a solid knowledge of how we do drug design at that point, and I just didn't find a way how we put these things together. The way I solve it now is by looking at the data we have. The data we have about molecules defines how we can model them. One way we can model a molecule is simply by its coordinates. So this is probably the most basic data format that we have for molecules. It's the XYZ file. The syntax is at the top is the number of atoms in the XYZ file. Below that is a comment field, usually used for the name of the molecule. I'm doing aspirin here. This is the structure of aspirin if you've not seen it before. And this is followed by a line-by-line list of the atoms with their coordinates. So an atomic symbol at the very left, cross-reference to the periodic system, which is a reasonably global thing, as well as then XYZ coordinates. The white space is not significant. The decimals aren't necessarily standardized in any way. This is a very, very rough file format. In fact, there are several things missing. So if you're paying if you're very sharp-eyed, you might have noticed that I have a whole atom here that is not represented in there. So all of this is carbons and oxygens. There's not a single hydrogen in here, even though I've even explicitly shown one here. There are a lot more hydrogens on this molecule that I'm implicitly abstracting out for chemical convention. Hydrogens are often left out of any file format because hydrogens are usually computed by whatever computational chemistry engine you are currently working with, which can have its difficulties when these engines do not agree. And furthermore, what we're also missing is all kinds of connectivity information. This is really just the coordinates, and we have no idea how these atoms are connected. And this kind of betrays where this file is used. This file format is mainly used in quantum chemical applications. In quantum chemistry, on a quantum chemistry level, on a quantum mechanical level, there are no bonds between atoms. There are only electronic distributions. And so when you put this kind of a file into a quantum chemistry engine, it will make its own bonds. This is useful in a quantum chemistry context, but if I take this file and go to a normal chemist with it, he will come back to me and ask me what the hell I am thinking, and that he cannot work with a molecule that has no bonds. The usual chemist works with bonds, and so bonds are a very important part. So let's see the other extreme in a file format. Yes?
Question. So back to
Yes.
the last slide. So all the numbers that came from measurement or calculation?
So this is actually a measured one. So all of this is measurement and calculation. This is a set of coordinates from a crystal structure that measured aspirin bound another protein. I've taken out the aspirin here. So it's scattering patterns of the X-ray that are then computed into electron densities that suggest atoms are in a particular position.
In crystal state, so is the structure the same from its soluble state?
Very good question. We will come to that. For now, we're going to look at static structures. I will have some dynamics later. But let's move on to the other extreme in the file format context of we have only coordinates to we have no coordinates and only bonding information. So this is the simplified molecular input line entry system, the SMILES. I'm not sure if you smile when you look at that. I don't think I do. This is a graph format. It's a line graph format, and the white atoms in this line format are suggesting the main chain of the molecule, the main chain starting from here and ending here. So we're going here, along this, and here. Now there are a few more features of this molecule that are not part of the main chain and that we have to express somehow, such as, for example, these branches here. So this double bond to an oxygen, and we do that with the brackets here. So this is how we try to make this a real graph and not just a line. And then there is this ring here. And so somehow we have to define the ring. And we define the ring through these sorts of connections across multiple entries or multiple tokens in the line. So here, this carbon is this carbon. I'm putting a 1 there, and then this carbon is this carbon. I'm putting a 1 there, suggesting this is a connected structure. This is how we go. We have graph formats for our molecules because, well, we do have nodes and edges. An easy way to model molecules is the graph. What else here? Yes, there is a very intricate and very convention defined structural element in here, which is once again this ring. So you see all of the we start the main line with uppercase C, which suggests a carbon. But then the ring is suddenly in lowercase c's. What is a lowercase c? It is also a carbon. It is an aromatic carbon. Aromaticity is a chemical property where you have a rapid exchange of electrons between atoms. And this is a very, very important feature for a lot of chemists. Without going into too much detail about what it means, But here the lowercase c suggests that. And this is also something that, depending on the chemistry engine, can be interpreted differently. And so sometimes when you load an aromatic structure generated by one chemistry engine into another, it breaks. So this is very unfortunate sometimes. And so in this case, the aromaticity is well defined. We have a very formal scientific definition that this is definitely an aromatic structure, as shown. If we had a nitrogen in there, the formal definition breaks down completely. And there are many different and disagreeing conventions on how to handle that, which means this case of chemistry engines interpreting things differently is actually very common. So we have this constant interplay of implementation and the scientific background, often fighting each other and leading to problems.
Regarding this slide and before,
Yeah.
before slide, you have mentioned the coordinates.
Yeah.
So, but where is the like? You have mentioned like 12 point something, and minus 74 something. But what is the like origin point like?
So the origin point for these particular coordinates was defined crystallographically by the measurement of the whole molecule. So this is a part of the crystallographic measurement, and the origin is defined by something else, not by this molecule particularly. Otherwise, yes, I could transform this into origin and it would be fine.
Okay.
Yeah. So this is a combination of both connectivity and coordinates. Once again, you have the coordinate block that you may be aware, or that you may recognize with all the carbons and oxygens and their coordinates. And then below that, you have the bonding information, which is really displayed as an adjacent. It's not an adjacency matrix, it's just a list of adjacencies. And this is probably the file format we deal with most commonly. Once again, at the top, we have a name field, and we have a field for the software that generated it. We have a blank line, which I forgot what you put in there, but it's not often used. Below that is a sort of metadata thing of we have 13 atoms in this file and 13 bonds. You may recognize that this is an 80-character line limited format because this is really a thing from the 80s, and we still are we are still using it very enthusiastically, though it has massive limitations, and this is why you also have this metadata information of 13 atoms and everything. There are a lot of zeros, both after the atoms and the bonds, and this is also various kinds of annotation for the atoms or the bonds. The one kind of annotation I will explain is here, right next to the atom index. Let me actually do this on the computer. Can you see my, yes. So these first two columns are atom indices, or node indices, however you want to see it, and that define which atoms we're connecting. And then the column next to that is the order of the bond. Is this a single bond? A single bond has a chemical meaning of generally 2 electrons being involved in it, and then you have double bonds, which get a bit complicated, especially in the case of the aromaticity. But we're annotating the strength of the bond here effectively. And then at the very bottom of the file, you have a way to arbitrarily set key value records, which in this case is just the job ID of a particular model server generating this file. But effectively you can put whatever you want in it, and we do use this a lot. And so a typical operation involves taking this kind of file format and transforming it into a data frame where all of these key property values are then put into columns. So yeah, we put a lot of information into this kind of thing. And so here we have something that we can work with in a computer. We have a graph. We have things that we can put 3D into space. And this is how we put molecules in the computer. And there are a bunch of different databases for different applications that use, for example, this file format, but also use other file formats, for example, the Protein Data Bank, which does this then for proteins, which is a bit of a larger, generally, amount of data, and therefore requires a different file format. And we do have a significant amount of open resources available to us as computational chemists to just look at this, and you have access to all of this as well. And so to explain a little bit what kind of resources are available, let's just take a look at a gene. I would go from a gene and see if we have any kind of interesting molecules associated with it, any kinds of proteins associated with it. Does anyone have a favorite gene? Otherwise, I have a funny one. No? Then I will do a funny one. Unfortunately, biologists have a sense of humor sometimes. And so we have something called the sonic hedgehog protein. The sonic hedgehog protein is a protein involved in the hedgehog signaling pathway, a pathway involved in cancer. Did I not click it correctly? What is it doing? Yeah, there we go. So this is a protein involved in, I forget what kinds of cancer, but in general involved in cancer, and I have seen this internally be a target that people develop for. So what you're seeing here is the record from UniProt. UniProt is a gene database. It associates functional genes with their sequence and their proteins, and this is usually what we map a lot of things through. So if I, for example, take this particular code and go to the protein data bank, which I believe its name describes its function. It is a data bank of proteins and specifically of 3D measurements of particular proteins. I can go and see, there are multiple measurements of this particular protein. I can click on one and we can see a 3D structure of this particular protein. So this will be saved a little bit different than the file formats I showed you, but this is effectively the structure of the protein. Okay, so we have the structure of a protein, we have a gene, we have some information associated with the gene that points to disease information. And the same way that I went into the protein data bank here, I can also go into the ChemBall, a repository of chemicals associated, or chemicals of biological interest in some way. These are molecules that are associated with some kind of bioactivity, with some kind of activity on proteins. And I can use the same code from the UniProt to go in here and look at if we have any particular measurements for this protein. This will take a moment, and I hope it's going to load. Yes, so we found our sonic hedgehog protein. And then I'm going to find the activities. This is the assays. Let's look at some associated compounds for this particular protein. And here we go. So we have a number of compounds for this particular protein. These are generally compounds extracted from literature, that is scientific literature. I believe the ChemBall does patents as well. I'm not sure if this is shown here, but this is the source for that information. And underlying this, if I go, so I know we have an approved drug here. So this is an actual drug that is on the market. If I go into the approved drug, then first of all I can see the smiles over here. I can see a few other identifiers, which I'm not going to explain. And I can also, okay, I can't show you it because it's a bit big. Yeah, but I can roughly show you a file format that you once again recognize, the mole file format, and we have some a little bit of metadata here. Effectively, I could, from the ChemBall, extract an SDF file, an SDF that contains all the data that is on this page, or most of it anyway. So at some point we will have the actual measurements. What is this? Oh, this is indications, okay. So this is a little bit different. Yeah. So here we start having bioactivities. This compound is not just measured against the protein it was developed against, but against many others. So we have a lot of information, and technically you could start doing drug design with this. You could connect all these databases, start looking through different things, look for a target that interests you, look for a disease that is associated, or maybe you probably start with a disease, see what kind of disease you want to work in. Do you have targets for that disease? Do you have compounds for that disease? Can you start a drug design program with this? Yes, what else did I want to do here? There's one thing that, and you will need something to tie this all together on a software level, and that is usually the RD kit. The RD kit stands for the Rational Discovery Kit, which is the foremost open source cheminformatics software that we have. It is dominantly used in basically every pharma. Yeah, it's open source. You can contribute to it yourself. I have its mixture of C++ on the algorithm side and Python on the more accessible side. Okay, let's move on. I think I'm taking quite a bit of time. Let's do some mathematical modeling. My doctoral advisor had the tradition of always starting his drug design courses with this particular quote that says, "Every attempt to refer chemical questions to mathematical doctrines must be considered now and always profoundly irrational, as being contrary to the nature of the phenomenon." And this is a quote from Auguste Comte in 1842. Auguste Comte was a positivist mathematician who obviously disagrees with using math in chemistry. As chemists, we are going to disagree with him because obviously he has no idea what he's talking about when it comes to chemistry, even though as chemists we usually don't have much of an idea about math. And so I'm just going to do a little bit of an exercise with you where we train a neural network and predict a property of a molecule, specifically the binding efficiency of a molecule. So I'm going to be working off of a Jupyter Notebook coming from Teach OpenCAD, which is a program to openly teach computer-aided drug design techniques. That's what CAD stands for. And this is effectively a Jupyter Notebook where they give you some theory and some practical applications in how to actually do this. While I'm not going to read through all of this, so this is a basic introduction into neural networks, and all of this is it is a bit dated, but we do still do these sorts of neural network things, so you can talk a little bit about loss functions and all of that. We're using TensorFlow, the old TensorFlow, I believe. And what we're actually going to do, I think that was up there actually. Let's see where it was. What we're actually going to do is we're working on an EGFR kinase. EGFR is associated with non-small cell lung carcinoma, which effectively involves, this is a lot of smokers getting cancer. And this is not just so this is usually not going for the EGFR kinase is usually not a first-line treatment, it's a second-line treatment. The first-line treatment is to try and cut it out and then do some chemo. If that still doesn't work, then we start going for things that hit the EGFR kinase, for example. And what we're going to be doing in this example is trying to predict the IC50 of particular compounds for the EGFR kinase. The IC50 stands for the inhibitory concentration, the 50% inhibitory concentration in this case. So what does it take to inhibit this protein to 50% of its efficiency? The IC50 is a value that moves exponentially. And it's also a value that is good when it gets smaller. So the less you need of a substance to reach the 50% inhibition thing, the better. And because that's kind of unintuitive for people, what we often do is we turn it around and make it a PIC50. The P is here standing for a negative decadic logarithm. So this is the same P as in pH. Effectively, the PIC50, the bigger it gets, the better. We are in significantly smaller numbers working linearly. So we're predicting the PIC50, and we're going to be doing that using, once again, Campbell data. So this is really just Campbell data extracted for the EGFR kinase. We have a little bit of a schema of what that looks like. You have some values here, actually. And here you can also see the exponential nature of the IC50 moving around. So this is a bad compound, big number bad, small number good. But then we turn it around with PIC50, where big number good, and everything works a little bit more linearly. We have to somehow encode this molecule for a statistical model, for a neural network in this case, and this is not trivial. We are working with graphs here. How do you make a graph into something that a neural network understands? And I have a slide for that because it's a bit of a mess. What you do is you take graph features and you use them as categories. So what we do is we make structural molecular fingerprints. These are long byte strings, bit strings, mostly bit strings, of a lot of zeros, and then once in a while you get a 1. And this 1 is associated with a particular structural feature. So for example, here we've detected this particular structural feature in this molecule, so we set this particular position as 1. Here we have 2 structural features, so we set 2 1s. And this is how we can, and this is effectively then categorical data that we can feed into a statistical model to associate a continuous number, in this case the IC50, with a category that is the absence or presence of a particular structure, to teach it to predict the efficiency of a particular molecule binding to a target. And this is what we do here. There's a definition of the neural network, there's some training stuff, different batch sizes if you're into that sort of thing. So if you are into this sort of statistical modeling stuff, you can take a look. This is going to be fairly simple. There are more sophisticated ones, but in the end, here, so this is model evaluation. We have a few values trying to describe how good this model is working. I'm going to focus on the last one, the MAE. The MAE is the mean absolute error. The mean absolute error in PIC50, so we are at 0.84 PIC50 mean error, which, considering that we are in logarithmic space, means we are about 10-fold up, 10-fold down, roughly, in error. To drag that abstract number into something real, that means, because this is the efficiency of the molecule working on the target, this is 10-fold up, 10-fold down, how much of the substance you will have to put in the pill to influence the patient's health. So this is quite a big error. And in fact, in context, this is actually a very, very good model. 10-fold up, 10-fold down is really at the state of the art. I don't quite believe that this is really that good of a model. But this is a very, very hard problem for us, doing this sort of efficiency analysis, or this binding efficiency, binding affinity prediction. And so 10-fold up, 10-fold down is actually pretty good. Which means it is a very hard problem for us because we still need it to get a lot better to be really practically useful. I'm going to leave that there. Here's some retrospective validation. So all of these points are different molecules that you repredict and then see, okay, this is predicted value, this is true value. And then you can also do the analysis of what kind of structural features are we actually looking at? What are the feature importances here? This isn't necessarily feature importances, I think. I think this is just qualitative analysis of the things that are kind of working well. But these are real chemical features that you can go to a chemist and say, this is something that's associated with activity in this particular thing. Maybe we want to do more of that. And this is something nice that you can do with this kind of a featureization, that you can say it's this feature that's really driving the activity. Okay, what do I have here? Let's do some structure-based molecule modeling. So this is the 3D stuff. We have 3D structures of molecules. We have 3D structures of protein. Can we just put them together? Can we see if they fit into each other? So let's just do that. Actually, let's skip the docking and let's go to an MD simulation. So once again, I'm using Teach OpenCAD stuff, and what I have here is an analysis of a molecular dynamics simulation. Molecular dynamics tries to do what we talked about before, which is not just have a static image of the molecule, but have an have a movie or a simulation of a molecule in solvent in the context that it's supposed to be in. In this case, we're going to do this in the receptor and only slightly exposed to solvent. But this is the kind of simulation we're doing here. We're doing dynamics, and this is a Newtonian approximation of molecular movement. So all of these are like physical springs and all of that. None of this is quantum chemistry yet. If you did this on a quantum chemistry level, it would take forever. I don't know if, I think we've done, we've done systems of the size I'm going to show you on a quantum chemical level. But these sorts of things take forever. And so the kinds of things that we want to do here, so this is a wiggling molecule. This is always very nice. I would be very remiss in a computational chemistry talk if I didn't show you a wiggling molecule. Let's look at ours. So this is once again the EGFR kinase. Let's take a look. We've simulated this. I've simulated this before because it takes a little bit. And what I can do is I can just play the simulation and we can take a look at this. So what we have, yes,
question,
yes.
Could you tell me the model?
Mm-hmm.
Basically, we can use the model of the molecule
Mm-hmm.
to the picture,
Mm-hmm.
right?
Yes.
Oh.
So yeah, so these are this. I'm not completely sure where they got the structure from. Usually, you could once again go to the protein data bank. You have a measured small molecule in a protein, and you can just simulate this and see how does this static image that was recorded by the X-ray crystallography or whatever other technique you used look if you started moving it, if you gave it solvent and all of that. And the reason that we do this is to see how comfortable is the molecule actually in its receptor and what parts are more or less comfortable. Comfortable is a very unscientific, difficult term here, but we can take a little bit of a look here and just see that. Can I? Yeah, I'm doing this on touchpad. I apologize, but what you can see here is that different parts of the molecule are moving different amounts. So the this part here, that was a mistake. This part here, for example, is moving a lot more and flipping than this part. And this is a very, very core binding motif for this particular protein. This is something that a lot of molecules in this context have. This is very free-form and also very solvent exposed. I'm not showing all the water around the protein. I'm not showing you a lot of things right now. So that you can actually see something. But this is mostly interacting with the solvent and a little bit with things up here. Yes?
So at this stage, are you doing like the electrical interaction as well, or is this pure physical? I guess at this level, I think everything is electrical, right?
So I don't have the classic slide of giving you all of the potentials that we have here. But the main driving forces here are, first of all, the sterics. All of these are basically spheres moving in space, bonded together. And so there's just sterics going around. But there is also electronics, such as a fairly simple Coulomb potential between all of these things. So all of these atoms have different charges, right? And nothing in this particular molecule is explicitly positively or negatively charged. This is something we usually avoid in development, to have that sort of thing, because it makes it very difficult to get it into the person. But you have partial charges, and these partial charges do drive movement. And I mean, water is partially charged already, so you have electronic potential between all of these molecules. Now, actually, let's go into that a bit more. So beyond just looking at the structure, we do metrics about it. This is an RMSD of the protein and ligand, root mean squared deviation. That is, how much is the protein moving? How much is the ligand moving? If the ligand is moving too much, then that is a hint that the ligand is not very happy here. And yeah. Let me show you this with a little bit more detail. So this is the ligand, once again, in the middle here. And then we have a lot of the amino acids of the protein trying to interact with the ligand. And one of the classical interactions for this particular molecule is this thing here. So this is a nitrogen and a hydrogen, and this is a fairly strongly partially charged structure, which is interacting with the nitrogen on the side of the ligand here. And what we can do is we can then analyze that particular interaction over the course of the simulation, and we can see it is very, very consistent. It is very, very consistent in distance between 3.5 and 3 angstrom, or 35 and 30 nanometers, I believe. So we have a very, very consistent distance for this thing. This is a very strong interaction and a very driving part of this. And you can do more statistics about this and do all kinds of other interaction analyses. So this is something we do dynamically. And then I think I should move on before I run out of time. So I thought I should do something about AlphaFold just because this is probably the one thing that you know maybe about drug discovery and computing and drug discovery, or structure prediction in general. So AlphaFold got a Nobel Prize in chemistry, and that was a very controversial issue, especially in pharma, because yes, structure prediction is nice, and let me unequivocally state that AlphaFold's techniques for structure prediction are an objective step forward, and the way they validated it and everything was super clean, so much more clean than a lot of the other AI initiatives that you see around. So AlphaFold is an objective step forward, but, and here I'm not speaking from myself, but from a colleague. I don't know if you know Derek Lowe. Once in a while, one of his articles makes it on Hacker News, for example. He has a fairly prominent blog where he talks about pharma, and in his words, it is very, very rare for knowledge of a protein structure to be any sort of rate-limiting step in a drug discovery project. Now, none of the techniques we discussed before are possible without a structure. That is explicitly true, but there are often ways to getting a structure or using just a similar structure that mean that it's very rare that this really becomes a problem. And so AlphaFold is a very, very good structure prediction tool and is useful, but it's not the revolution. It's not going to revolutionize pharma as much as people maybe want it to. And now I shall move on to computing techniques. So this is maybe the part that's more interesting to you. I will talk a little bit about how we use the HPC and things like that. And so starting with, we are very, very heavy users of on-premise compute clusters. I've always had an on-premise compute cluster in every organization I've worked at, that is both of the universities and Novartis and all of the pharmas that I occasionally worked with. Which are usually on a grid engine queuing system still. This is very unfortunate. You know, you'd hope that they would go for something modern, but that's usually not the case. We all grow up on grid engine, and that's how we live. Startups tend to be more cloud focused. There was around, I don't know, I guess 2016 or so, a push for a lot of the software vendors to have some sort of direct connection to AWS, to various cloud vendors, to offload calculations that you start on your local machine into the cloud. And startups do need this sort of scaling that you, or want this kind of scaling instead of building their own on-premise cluster. There are enough pharmas that do these sorts of cloud experiments as well. A lot of pharmas do have some kind of cloud. I believe we had both AWS and Azure at my last job. I'm not sure who was using what. I'm not sure if I was supposed to be using any of them. We always buy everything in triplicate in industry somehow. What the cloud focus really shows you is that at some point, some of the more computationally demanding techniques that we work with start approaching costs for experiment. So, for example, very, very sophisticated molecular dynamics simulations do start to hit costs of why don't we just synthesize the molecule and put it into a physical assay having a significantly stronger physical readout than any simulation ever could. And so this is one of the interesting results coming from the sort of cloud focus, that you do start to see computational experiment costs approach physical experiment costs, and how you have to start weighing those against each other. you are usually sharing these resources with users of very, very variable backgrounds. So I think I have a thing about backgrounds in a moment. But for example, one thing I've talked with Brian about quite a few times is that how much we use containerization, which is very, very little. A lot of the people working in computational chemistry write a script and just want to put it on the cluster somehow. They don't care. They are not learning Docker. They are not learning Singularity. They are not learning any of this. They just want to Q-sub it somehow. So containerization tends to be limited to expert users. You usually have infrastructure running on containers, maybe coming from directly IT, maybe coming from shadow IT. I have another slide on that. But it is a lot of just various dirty scripting, and you hope it's just Python or just Bash, and not someone insisting on Perl or something like that. There are usually also many, many different systems that abstract the job submission process. A lot of these vendors have their own job queuing systems that they try to make their software more user-friendly with by abstracting the job submission process away from the users, which can be a big pain on an IT level because you then have to somehow synchronize your queue names with the queue names in the vendor software and things like that, which leads to very strange situations and a lot of frustration from users who don't understand the underlying problems and don't care, and who have the luxury of not caring because they are experts in their field, and that's why the company hired them and not for the computational skills. So HPC environments, we do quite a bit of GPU acceleration, specifically molecular dynamics is a big thing that in structure-based modeling that does a lot of GPU stuff. Nowadays, a lot of GPU usage is dominated by computer vision and deep learning. Computer vision is something that you see a bit more in biological things, so you have a lot of cell counting and that sort of analysis going on a computer vision level, so it's a lot of the biologists then fighting us for the GPUs, and the deep learning people as well, or the general AI people trying to accelerate their learning with GPUs. GPU acceleration can be beneficial, but it has to be used appropriately. So you can't just GPU accelerate everything. People try. But yeah, there are some issues with this. So one of the more interesting ones is GPU code tends to use 4-byte floats, and that changes results. So you will have the CPU implementation and the GPU implementation of the same program, and because of the difference in float precision, it will deliver different results, which is very, very uncomfortable for a scientist. Because why should it do that? That doesn't make sense. And so if a scientist sees that, they are very, very nervous. The big thing that limits GPU acceleration is usually moving large amounts of data that can, and the movement process itself can outweigh any kind of speed ups you have in the operations that you do. I have an example in a moment. And lastly, someone does have to maintain GPU code, and we are definitely not doing that in pharmaceutical industry. We are not software industry, and so pharmas are not paying a guy to write CUDA. And so hopefully they can buy it, but then the question is, are you willing to pay the extra for buying GPU code, which is usually a premium? I have an example of something that someone tried to GPU accelerate. This is a talk from some people in NVIDIA that came to the RTKit user group meeting that talked about doing molecular fingerprint comparison. So we did the fingerprinting just now for input into statistical models. You can also compare molecules that way. And the two bars on the right are how fast the GPU code is, and then on the left, the four things there are state-of-the-art systems that we normally do fingerprint comparison with. What this graph does not show is that this speed up breaks down instantly when you have a certain amount of data that is a normal amount of data that you actually want to do this comparison on, and then it is really the moving of that data onto the GPU, which has a limited amount of VRAM that limits your processing of that data. So yes, you can do GPU acceleration often, but it's a very detailed thing of whether it's actually worth it or not. Yes?
Can I have another question?
Yeah.
So maybe this is coming more from the ML side of things, but we have a lot of like different data quantizations, right? So you can go all the way from like your floating point 16 all the way down to whatever tiny value you want. Can you use those sort of techniques maybe to kind of apply to your data, maybe and run different tiers of simulation, for example?
So yes, in this case, these are all bit strings.
Okay.
So there's no, so you couldn't shorten the float in any kind of way.
Okay.
There are, they did mention some memory reuse optimizations that I did not fully understand. So that's something that you could read, and I have the link there. You can take a look. I think they have the code online. So if you're interested in that specifically, I think you can try and read the CUDA. Yes, you can do some stuff. The thing is that even with optimizations, it's going to take an asymptotic difference, not like a factor 2 or a factor 5 to really make this viable on a GPU. Yeah. I thought I'd do some quantum computing. We do some quantum computing once in a while, or we try to. So emerging quantum computing may have applications in pharma as well. There are theoretical overlaps between the theory in quantum computing and the theory in quantum chemistry. They are both quantum, and you can do some shortcuts in between that. At the moment, these are just rarely pragmatically applicable to drug design programs. It's interesting to see that very few quantum computing papers actually measure runtime, and the reason for that is that there's just a lot of overhead, once again, like in the GPU example, to encode things into qubits and encode them in an error-corrected way. And so at the moment, this is not really a thing, but we do talk about it once in a while. Let's do some organizational challenges. Shadow IT is extremely prevalent. Most software reuse started in an academic environment, and that comes with a bunch of stuff like inflated complexity for publishability. The maintainability is probably not great. It's usually, or it's often very old, so I have had to deal with enough Fortran stuff. And there's often a disregard for conventions, that is conventions of language. What language are you using? Most of our users can barely mumble Python. If someone can do C++, they are already pretty good in the community. But then there are people doing OCaml. And no one's going to be able to read that. Like, it's nice that you can do that, and I really don't mind your choice of the language, but if no one can read your code, no one can analyze what's going wrong if it does go wrong, and that's very difficult, or this sort of thing. You need both significant skills in science and technology to maintain and test the sort of software. We have complexity in the technology, quite a lot of it. We have quite a lot of complex algorithms going on once in a while. We had the NVIDIA people doing CUDA stuff, and then there was immediately a guy who stood up, but this is memory limited, it's not, yeah. So you have people with that kind, you need people with that kind of skill set, and then you need people with a lot of a scientific insight into this sort of thing to actually see if any of these results mean anything. And that's fairly rare to find in single people, and if you split it up, it becomes a different kind of mess. This is what companies often try to do. Companies try and regret offshoring IT in cycles. So this is a phenomenon, I believe, in most IT industry, but in our case, this is exacerbated by the fact that we do need the scientific knowledge, and you cannot just explain to an IT contractor what the result is supposed to be for a molecular dynamics simulation to have been correct, or for the engine to have been correctly compiled. And then you have the test suite running, and then, I don't know, 5 tests fail. And does it mean something that these 5 tests fail? Is this a normal thing now? Do we accept that? That's a very difficult decision to make. Yeah. And then there's the computational chemists themselves, which are often not easy to deal with. So we have very varying expertise from very talented chemists who can maybe use the command line to very talented computer scientists which couldn't tell the difference between 2 chemicals if their life depended on it. And everything in between, which makes communication very difficult. Chemistry is in general not a very communicative science. We came from the alchemists, and unfortunately some of that tradition is stuck. But yeah, so this gets very difficult in communication, and also what the group consists of. More computer science-y sort of people or more chemistry sort of people dictates the environment that you're going in. This is the software environment, this is the way that things are communicated, and all of that, which can be, yeah, you have to find your place in that, which can be difficult. Then I have some takeaways from you. So this is computing and drug discovery is always a mix between scientific and technological skills. A lot of the technology is defined by the science, and the science is often unclear, or the technology is often clearer than the science. We have significant free resources available to us. Those are not necessarily Libre or anything. I haven't really been talking about the licenses. I believe our DKIT is BSD or MIT or something like that. So we have a lot of commercial use, and a lot of companies actually drive the development of these sorts of open source implementation. Our DKIT is to a significant degree the reference implementation for a lot of what we do in computational chemistry. Computations are varied. We have some GPU usage, we have a lot that's just CPU, and it's fine with just being CPU. We have a lot of users doing very weird things with their scripts. It's rarely cleanly containerized, and we do often need significant resources for this, which can be a challenge in a HPC environment. So with that, I've put some links in here. I'm happy to provide the slides if you want to take a look at this. So I extensively used the Teach OpenCAD stuff. I didn't get to use some of the stuff from my own lab, but this is the sort of, these are a few links that if you're interested in this sort of thing, you can definitely check out. I have my own GitHub and my ORCID. The ORCID is a record of my publications, so on one hand the technical, and on the other hand the scientific side of what I do, if you're interested in that, and I'm happy to take any questions. Yes, please.
I'm curious about the bitstream code. You
Yeah.
mentioned, for training, it's mostly zeros. Is there any compression going on?
So the usual compression. The usual compression for this sort of thing is you don't actually write the whole bit string. You just write the bit indices down in an int vector list. It is that sparse that this is already a significant compression. The reason, so you will at some point need the full bit string as an input for the statistical model. The statistical model tends to need a fixed length descriptor for a particular point. So if you, we did a neural network here, right? Or I did a neural network here. The neural network has a number of input nodes, and those are fixed. And there are ways around that. Yeah, you can do some dynamic stuff. Generally, you will have these things be fixed, and that's when you put out the whole bit string and turn these nodes to 0 or 1.
Is there any data about the correlation of the actual carbon chain length versus? It seems like it would correlate that closely.
It would what?
It seems like it wouldn't correlate that closely, but apparently it does.
So the issue is all of this is kind of categorical, right? Whether something binds to a protein can be defined by a single one of those bits. It can be a combination of a few of those bits to define the efficiency there, but if one of those structural features is missing, you can completely lose the binding, which it's not nicely continuous correlation. It's the sort of categorical mess of every one of these categories being able to have a very strong effect. You can also, yeah, please, you wanted to say something.
No, I mean, I mean, what you're saying makes perfect sense.
Yeah.
0 is clearly not going to bind, but a 1 just means possible, right?
Yes, it doesn't mean that that this structural feature necessarily influences anything at all. There are a lot of so this is a very handwritten kind of way of decomposing a graph into structural features. There are a lot of other ones, and then if you start just decomposing the graph by nodes or by neighborhoods of nodes, then you will have a lot of duplicate ones and things like that, which then can intercorrelate and things like that. And so some of the features in these bit strings usually are just white noise in the background. And there are quite a few publications analyzing, okay, which ones of these bits are actually in any way relevant, which ones should we take out to avoid noise and things like that. Yeah.
I mean, doesn't it all just depend on different confirmations of the molecule itself?
So we're not doing any 3D here, which is another thing. We're not doing any 3D information. This is just presence or absence of specific structural features. The conformational thing is another level above that, yes.
So then, before AI,
Mhm.
were people using this technique just mathematically by hand? Or-
So before AI is a difficult question. What these descriptors were originally used for was similarity search for different molecules. You would just do a union over difference over union, union over difference, union over difference. So a Tanimoto or a Jacquard index, or whichever is more familiar to you. This is what these descriptors are for, and then we started putting them into mathematical models, starting with just linear regressions to now the more sophisticated architectures. Please.
Sorry, another kind of ML-ish related question, but I think, is there any like experimenting or looking into like maybe moving molecules into latent spaces, for example, and then feeding those through your models instead?
Sorry.
Using latent spaces.
Latent spaces, yeah. So we do. Let me think about what we have on representation learning. Yes, we do have encoder-decoder things where we then take the latent space and start doing, and start using those as features. This exists. It hasn't shown a massive difference to some of the more automated decompositions of the graphs. And the big drawback to doing the sort of latent space stuff is that here I can point at nodes in the graph. And if I have to, I will at some point have to go into a room of chemists and say, this thing, I like this, we should do more of this. And then it's very nice to be able to point at a cluster of nodes to define that, and best of all, a structural feature that is chemically meaningful. Which is why in 2020 these people are still using this very handwritten categorical descriptor, because it's just very nice to point at a chemically meaningful feature and say, yeah, this is what we should do. So we do have some latent space stuff. Yes?
Yeah, after finding the structure of the compound, is there any guarantee that the compound can be synthesized?
Well, no. No, obviously no. That is the big conflict between any kind of. So we also have generative chemistry, generative AI for chemical molecules, and we have a few different architectures doing this. And the problem with the molecules that are spit out by that is that we have no guarantee if this can be synthesized, or more importantly, if this can be synthesized in an economically reasonable way. So the typical workaround you have for this sort of thing is you do whatever generative chemistry, and then you search against either a database of vendor molecules that you can just straight buy, or you go into combinatorial synthesis spaces. Okay, I don't have the. So combinatorial synthesis spaces where people take a bunch of reagents on one side, a bunch of reagents on the other side, and simple reactions in between, and generate a huge space of potentially synthesizable molecules. How realistically synthesizable they are depends on how solid the reactions in between are understood from like environments and things like that. There are a few prominent companies that will give you not guarantees, but will tell you, okay, with like an 80% chance we can make that molecule. And so often enough you do the generative chemistry stuff, and then you go to one of those huge spaces with potential molecules, say I want this one, this one, and this one. The vendor tells you, yeah, we can do like 8 out of 10 of those, and then maybe you only get 7. because something still went wrong along the way. So this is a constant risk, and this is why you rarely go for one molecule to synthesize. You synthesize a few and then see that you don't lose too much along the way, either through it being unsynthesizable or then the prediction just being wrong and it's just not binding. Yeah, scary stuff.
Any other questions?
Yeah. Yes, so you
Mhm.
mentioned the MAE result is 0.84, and
Mhm.
it's, even though the result is not good enough, but it's a great model. So my question
Mhm.
is, why could we trust the model since the
Mhm.
result is not okay?
So the result is not good in the sense of a model with that kind of error is difficult to use in practice because an absolute error in the logarithmic space that we're in there, that is roughly tenfold up, tenfold down, means you might have to put 10 times more of the substance into the pill. And so this is the pragmatic thing of the error is not really practical to you, or it's not practical. I understand the theoretical question you're asking of if it's that bad in reality, then why should we trust it?
Yes.
So this is a very difficult topic, and I made the slide for it this morning, and then I decided not to do it because it's a very controversial topic. I believe we have not solved this problem, but there are many people making a lot of money with the knowledge that we have solved this problem. And so, no, I do not believe that model. I do not believe that model. It is far too simple. It had no three-dimensionality, for example. That's a factor that it just didn't have. It had this very, very simple featurization. The only way, or the way I believe we should validate these models is through through truly prospective benchmarks. We have a few of those. What they do is people make their models. They can make their models however they want. They can make them physical, they can make them AI, they can make them whatever statistical modeling they want to do. A company donates a proprietary data set that no one outside the company has seen to an organization running this benchmark. The organization sends out the structures of the molecules and the structure of the protein, for example, for everyone to say, okay, this molecule is better than this molecule.
Yeah.
They send all of that back to this organization, which has not revealed which molecules are better. And then at the end, that organization writes a paper and says, these predictions were good, these predictions were good. It's like the CASP challenge, if you're aware. The CASP challenge was what propelled AlphaFold. So it's a very nice prospective benchmark. This prospective benchmark, these kinds of prospective benchmarks regularly show that our models, you know what? No, now I'm going to do it. Now I'm going to do it. Here we go. This is CASP 16. CASP 16 recently did this. These particular benchmarks tend to show that we have very, very big difficulties outperforming very stupid models to the point where it's difficult to say if we're learning anything at all. So this is CASP 16. CASP 16 did this sort of a benchmark of can we tell which molecules are better than others? And the result was that it's interesting to compare this experience with the previous experiences. This sort of benchmarking has been going on since at least 2016. So the D3R Grand Challenge was the one before that. And since 2016, it is not clear that the this is the post-prediction, sorry. So the post-prediction is another thing. In general, there do not seem to have been significant methodological improvements since 2016, which is a very damning statement. On the right here, I have the graphic showing the best model in this particular analysis. This isn't quite a Kendall's tau, but it's getting there. So we have roughly a Kendall's tau of maybe 0.4, and next to that is a model that only considers the molecular weight of the molecule. The molecular weight of the molecule is we are basically just counting the atoms. This is not a thing that makes sense. This should not predict anything. And yet it is almost outperforming the best model, and it is in fact outperforming more than 90% of the models in this benchmark, which is a very difficult reality. So I do not believe that we have affinity ranking, but we need it. We need to be able to tell what molecules are better, and so we try all kinds of stuff. Yeah.
So you're comparing with other models for what
Yeah.
reality. Is it better than random? If you're going to...
So the molecular weight isn't random, but it is a baseline model. So what we're doing here is we have a real data set. We have a real data set that's hidden, and all the different people participating in this benchmark are sending predictions to a central organization that.
No, I
Yeah.
get the
Okay.
test.
Mm-hmm. Okay.
My point is, when you're making a model, is it better than what you were doing before?
Mm-hmm. Mm-hmm. So this runs into more of a social problem than a scientific one. How are you going to measure this in a real project? In a real project,
Cost.
yes, but then so let's set up the experiment. The experiment is we do what we do anyway. And we try to optimize a molecule to have the best affinity so this is hit to lead. This would be the hit to lead process where we get the most affinity. Unfortunately, even in hit to lead, we will, you weren't there for that, I did a little bit of a thing. We will do other optimizations for the molecule except the binding efficiency. For example, we need it to be soluble in water, and if it's not soluble in water, then we don't care how much it binds. So there's a little bit of a mess going on there that it's not one objective necessarily. But then we also need to set up the other arm, and the other arm would be purely computational. And so you would have to convince an organization to put a very, very significant amount of money on that sort of thing, which it happens. It happens. It does happen. It happens very rarely. I know of a few cases where they really just trust the model and just run the model, and they run it directly into the ground from most experiences. Because of, for example, things that we are not outperforming baseline models, and you see that in these sorts of situations. And we are, as humans, looking at these molecules. We don't have the numbers. No, I can't really claim that. I can't really claim that we as humans, looking at these molecules and having all of the data that we have, can make better decisions about the molecules than a random model. But
Speed is important there too.
Speed is-
So if you're gonna run
yeah.
actual physical experiments.
Mhm.
Where's your starting point? Maybe you should start with this is plus or minus 10, and then actual test this.
So maybe let's put this on a bigger scale. On a bigger scale, there are companies trying to do very computationally focused workflows versus what we do in pharma, which is still very, very defined by old school chemists doing old school chemist things. And so you will have companies like Isomorphic, which is the spin out of DeepMind for AlphaFold, that do very, very computationally focused workflows where they do endless computational cycles before they synthesize one molecule or 10. And until now, Isomorphic isn't even the first company to do this. We had computational techniques before, and so something like Schrödinger, which I don't know if people have heard of, they are being called an AI company these days, which is very funny. But they also did they were very invested in structure-based modeling, and through structure-based modeling techniques would do endless computational cycles before they would synthesize one molecule. And the experiences from this have been mixed. The models are, without being able to point at one particular paper to say that this is the comparison of these two arms and this arm worked better, the experiences have been that you cannot just run the models. It does not work. The model
What
does.
do you instead?
You do a combination of having the computational people talk to the chemists and have the political discussion of trying to get the chemists away from doing a random walk in space and guiding them by the models that you are running, which is the obvious thing to a significant degree. And then the question is, how much do we trust the models versus how much do we believe in chemists and their magical intuition? You have different company cultures around this. I come from a very computational direction, and I'm already quite conservative when it comes to how hard we run just models. There are other company cultures, and whether those persists are, I believe, something that has to be tested by history, because it's just not working out to get companies to. We had in Novartis, we had several people very, very clearly suggest, let's run two of these arms in parallel. Let's just do it. Let's just see it. But no one wants to do that because it's a significant thing of resources that can lead to explicit failure.
Well, I mean, also the models, just looking at what you've shown, it
Yeah.
seems bad.
So, I mean, I'm not showing you state-of-the-art ones.
Well, it seems like there's obvious improvement everywhere.
There is obviously needed improvement
Well,
everywhere.
not just needed, it's like
Yeah.
I could fix a lot of it right away.
Yeah, I mean, so the neural network one is a very, very simple one. There are a few ones that are a bit more sophisticated and where it's then a little bit more state of the art. This is a teaching Jupyter Notebook.
But the
This
bitstream
one-
concept is still common, right?
Hmm?
The bitstream concept is still common, right?
The bitstring concept is very common. There are other featureizations. Do I have? I don't have it open. One other thing that we do, which is maybe a bit more interesting, especially in an AI context, is graph neural networks. If I was going to do some kind of deep learning molecular property prediction thing, I would go from graph neural network, where you have the graph learn a representation on a graph level, which is a lot nicer, or is a lot more flexible than these sorts of decompositions. This is what I would believe is the state of the art here. The problem with all of these models is that at some point we're data limited. You can run a graph neural network organization-wide because you have the data points for it in a single project. And in a single project is where you would do the affinity prediction, for example, because you can't do that across systems, really. The reality is you do not have more than 10,000 data points. Like I had access to the size of projects towards my later time at Novartis, and there were like maybe 5 projects that had more than 10,000 data points in a 10-year time span. And there you're still pretty well off with just classical modeling stuff. It's just not worth it to spin up a neural network. It's difficult. Okay, I'm taking a lot of time. At some point, someone will have to stop me.
Any other questions?
Yeah. Talking about the setting of servers,
Yeah.
about GPU, I think we got a lot of quality of GPU to prepare, right?
We have a lot of
We have a lot of
Quantity.
quality
Qu-
of GPU.
Mhm.
I think solid line is a little bit expensive to
Mhm, mhm.
get that, right?
Yeah. Yeah, and this is very much a question in GPU acceleration. Is it worth the investment in the GPUs financially to achieve that speed up versus CPU?
For example, like NVIDIA
Mhm.
or something like company,
Yeah.
we should prepare for that realize.
Mm-hmm. Yeah.
So
Okay.
basically, alchemy with some numbers and intuition.
The reason I invoked alchemy is because people were intentionally obfuscating stuff, and chemists are still very bad at communicating things, even if they're not necessarily intentional about it. Yeah. Okay. Then I will. Okay, well, thank you very much. I will give over to the next person.